Hi everyone. So I haven't made a video actually in quite a while, so I'm gonna try to get some up. I also have about a dozen questions in the backlog that I have gotten that I also have to make videos for. I'm at ECSS right now in Prague, and I'm presenting this poster that you see in front of you. So I thought this is a good video to make to show kind of people what I've been doing, this is a lot on this poster, so I'm gonna actually split it into two videos. So this is gonna be video one, this is gonna be a second video. The premise of this poster is kind of reliability and validity of NIRS of muscle oxygenation using NIRS on a 0 to 100% scale. It's something I've talked about before, and I'm presenting a test battery, which you see here, these 5 tests that help you gauge your NIRS device, in this case the Moxy. So we're going to go through these tests and the results. So we're going to look at test 1, 2, and 3 in this video, and those are reliability tests, so repeatability and reproducibility. Repeatability is repeating your test on 2 different occasions with all the conditions being the same, and in reproducibility you change certain conditions, and that's what we did. And in order to do this, I recommend in the test battery, because I think it's a stable environment, is if you do an arterial occlusion, which you see this leg, you see this pressure cuff on the leg right here. So you occlude the leg, so there's no blood flow. And we did put 4 NIRS monitors, so 4 Moxy monitors on the leg. And what you end up getting is this kind of graph over here. We're going to zoom in on that a bit. After 6 minutes of the occlusion, you plateau out your SMO2 at the bottom, you get a min value, and when you open up the cuff, you get a max value. And so the whole analysis has to do with this repeatability and reproducibility of these min and max values. And so what we ended up doing is we did passive occlusions, so the participants would just lie down, and you do the occlusion, and you do it again a week later. That's how to test repeatability. And then we also added the active condition. So it's the same occluded limb, but now instead of just lying there, the participants did knee extensions. And this adds another component for the reproducibility part. And if we kind of go back up to this test, we see when we say reproducibility, we want to check if this min and this max value, if this range is reproducible between individuals, between muscle sites, so all the different muscle sites we're measuring, and between a passive condition and an active condition. If this min and max value, or this range that we have available to SMO2 is really there, it should actually be there in everyone, in every muscle, and regardless of activation level, because the limb's occluded, so you should get this minimum value, this minimally oxygenated state, and this maximally oxygenated state regardless. That's the prediction. So we ran that, and these are actually, quickly take a look at these results here. So we'll go over to this results screen here first. And what we have is we're going to take a look at these Moxy passive trials SMO2 min values. These are the SMO2 min values for the 4 muscle sites that we measured, and they're the means of all the participants. And what we end up seeing is that they're all actually pretty close together. They're statistically not different. The average of all 4 of these is actually this dotted line. It's about 10%. So regardless of the muscle site, they all come down to the same. They all come down to the same value during the passive trials. The same is true for the SMO2 max. They all kind of come up to the same. So this dotted line again, it's about 80%, and this is where they all get back up to. And they are all statistically equal as well. So that all looks very nice as expected. The active trial min here and the active trial max, again, it's the same thing, but this time we added activity. So with the activity, you end up getting for the vastus lateralis and the vastus medialis, these VL, VM right here, you get the same type of value, this down to 10%. The RF was actually a little bit, let's say, out of whack. It's higher with a lot more variation. These two VLLVM were statistically equivalent also to these other passive MINs and to each other, but this RF isn't. There's just too much variation. Not quite sure why that ended up being. I think it's an experimental problem that we had with the occlusion and with the activity, but just to note that's there. For the MAX trials, we generally had a little bit more variation in the hyperthermic response. So in the MAXes for passive and active trials, but they're all not statistically different. So those all fulfill the criteria that we'd set up. So this kind of fulfills this. These different muscle sites fulfill the reproducibility, and the reproducibility between muscle sites and the active-passive reproducibility is also confirmed. Now, if we kind of scooch over here, these are the mean differences, okay? And so if we look at these passive trial MINs right here, it's the difference between my first passive trial, and my second passive trial. And so you'd want, if they're repeatable, you want it all to be 0, so there's no difference. And that is essentially what we get. Important now is that you have this top and bottom dotted line. Those are what are my equivalency interval. So I want my data to be in between here, and then my a priori assumption is then they're equal. And they're all in here nice and equal. So my trials are repeatable. And that's true for my min and actually also my max values. Now you have a lot of variation here. So again, this RF value here is a little bit of too much range, but it's statistically speaking still not different. So it all works out all right. And so the sample, if you have more sample size, this might come down. For the active trial here difference, these two, what's important to note here is that this isn't the difference between an active trial 1 and 2, because there's only one active trial. This is the difference between the passive trials and the active trials. And I'm assuming that there again should be the same. And that's what we see in the VL and the VM, right? They're in my equivalent interval, they're the same. But the RF isn't, which goes back to this value we had over here, right? This larger range. So this RF value kind of goes out of whack. But for the max trials, they're nice, pretty close inside my equivalency interval, so they're equal. It all turns out pretty good. These two values here and the ones here that we see, those are comparing with the Portamon, which is a different NIRS device, which has the max values for the Portamon and the Moxy are pretty similar. But the min values are completely different. And I talk about this in a different video about just how NIRS devices vary. So that's important to note. But that's reproducibility and repeatability. So that's test 1, 2, and 3. And I will talk about test 4 and 5 in the next video.